This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
So good evening, everyone. Welcome to Science at the Theater's 7 big ideas. I'm sorry. You will understand, one of our speakers had a medical emergency and she cannot attend. But if this format succeeds, we'll likely do it again in the fall, so she will have her chance to do that. So that's Carolyn Larabelle. So we wish her well. As you know, we like to experiment with the formats at Science at the Theater, and I think tonight we probably pushed the limits. We are going to ask our now 7 Berkeley Lab scientists to educate, excite, and inspire you in just 8 minutes. Only 8 minutes. Now, contrary to popular opinion, scientists, are not necessarily secretive, somber, silent people. They actually can be quite chatty. So this will be a challenge. We assume that they can do it. We know that they can, but just in case, Ross, we have a timer. So, as a test this evening, I would like you at about the 7:45 minute mark just to applaud very softly. So let's practice. Practice. Soft applause. Maybe a little louder. Okay, that's good. Okay. And then that'll tell the speakers that they have just, 10 seconds or so to wrap it up. And then when they're done, you can applaud wildly. With that, I think that's probably all I need to say at this point, and I will now ask you to give a warm welcome to our first presenter, Mr. Blake Simmons, who's going to be talking about some big ideas with microbes.
I'm not used to the timer running up, so please bear with me. And I also want to take just a moment aside. I can't believe you missed this PR moment to wish all the mothers in the audience belated Happy Mother's Day. That's a great filler, by the way, for future reference. Thank you very much for all that you do, and hopefully I won't embarrass mine tonight. And I hope you're watching, Mom. Okay, so the main message that I want to convey to you is that we are faced with some unprecedented challenges in terms of the carbon balance on the planet and also on the economic forces at play around energy and chemistry. And I wanted to start off this by giving you a perspective on what we talk about when we are conveying some messages about renewable energy and renewable fuels and chemicals. And instead of just focusing on one particular product, I wanted to give you an idea of what we face when we're really talking about replacing the whole barrel of oil. And the title of the talk was a little stilted. It's not replacing a barrel of oil, which we could probably do today, but replacing the whole barrel of oil for humanity to really enable this low carbon economy that you hear a lot about to address greenhouse gas emissions and other items. Exactly. So, but in order for a new low carbon bio-based economy to really become reality, it's not sufficient nor sustainable to just focus on one product. You really have to go after and understand that out of a single barrel of oil, you get diesel, other distillates, jet fuel, other products such as polymers, heavy fuel oil, which is kind of the residuals from a whole barrel of oil, liquefied petroleum gases, and of course, gasoline. And this requires an integrated systems approach. And it's something that the scientific community and industry really haven't addressed yet because it's typically, they only hear about a single point solution. And what you need is a system of system solutions to make that happen. And also to put it in perspective, and I've heard a lot of people around this neighborhood talk about, oh, it's just another Apollo space program or a moonshot and we'll be there. Not even close. The cost of oil imports, and this is not just from the United States, but China and the EU. In the United States, it was about $300 billion in 2010. That's 2 times the entire budget of the Apollo space program per year. In China, that's about $135 billion. That's 5 of the 3 gorgeous dams built each year. In the EU, about $300 billion, same as the United States. It's 20 times the Channel, which I don't think they have enough available coastline to actually make happen. And so, it's an enormous problem. And to think that we're going to achieve the end state in 24 to 36 months just isn't practical. But that also opens up the door to significant advances in science and technology to make it both affordable and truly sustainable. And the goals in greenhouse gas emission reductions in the state of California is certainly leading the way, so applause there as well. We live in one of the best states in the nation in terms of greenhouse gas emission reductions. And AB 32 set these really aggressive greenhouse gas targets to achieve 1990 levels by 2020, which from a science and technology perspective is right around the corner, and 80% below the 90 levels by 2050. And there are a lot of advances that can happen in terms of renewable liquid fuels, batteries, and fuel cells. And you have a bunch of different scenarios that have been postulated to meet that goal. And so you have light-duty vehicle scenarios, and I apologize for the slide being really small, but you have a bunch of different technology scenarios to help meet the greenhouse gas emission reduction targets for the state. And then nationally, there's a recent report called the Transportation Energy Futures, and I really encourage you to take a look at this report, that goes over the advanced fuels and advanced engines and technology sector crossover to make it happen. So there's a lot of hope out there, but there's not a lot of reality yet. And that's part of what we try to do at the place that I work at, the Joint Bioenergy Institute, is to help make the next transportation revolution happen sooner rather than later. And also to give you an idea of the baseline, these are the current estimates for renewable chemical production. So again, it's not just fuels, it's also chemicals. Here's the bio-related sales in terms of billions. And you have, you know, a whole list of products here in terms of like ethanol and biodiesel for biofuels. You have natural rubber, polylactic acid, and polyols. But you sum it all up, and it looks really impressive, right? $95.5 billion in annual sales. But you put that in the context of the $4.7 trillion that we spend in this market each year worldwide, and it is a pittance. And so you really need to be serious about it and appreciate the size of the problem before you can start addressing it. And building the sugar economy. A lot of what I'm going to talk about are products that can be derived from sugar realized from sustainable lignocellulosic biomass. And again, I'm not going to spend a lot of time on this, but I do want to convey the message that pretty much everything that we derive from a barrel of oil we can replace based on biologically derived processes. So it's not a question of if, it's a matter of how much and how soon. And that is a real message of inspiration for me every day I wake up, and that's what makes me go to work happy. And now, there's a lot of debate around food and fuel, or food versus fuel, in biomass and biofuels. I want to point out this USDA DOE study that was called affectionately the billion-ton study. And by telling you that, you probably understand the punchline already. But it projected that if you set aside food production, you set aside some of the conservation lands, and you really look at the deployment of agricultural resources and potential crops that could be dedicated for energy, it's about 1 billion tons by 2030. And so if you follow that through all the way to the chemical production element, 1 billion tons of this domestic sustainable biomass could produce about 1/3 of the current demand for transportation fuels by 2030. But only if we have the conversion technologies available to convert that product, convert that feedstock into a renewable endpoint. And so to do that, what you need is this integrated pipeline to take advantage of the natural terrestrial biomass feedstocks. And by this, we're talking about agricultural residues, hard softwoods, dedicated energy grasses. We're not talking about corn, and we're not necessarily talking about ethanol. At the place I work, we have several mantras that guide us in the day-to-day operations. First and foremost of those is that ethanol is for drinking and not for driving. And so what we're looking at is how can we take these natural feedstocks and convert them into drop-in replacement fuels for gasoline, diesel, and aviation. That's really where you get the impact. Because then you're not talking about the infrastructure concerns, you're not talking about $1 trillion to distribute a new product. You're utilizing the existing infrastructure to make those fuels happen. But first, you need to have the supply of those feedstocks. You need to really make the sugars from them, and then you need to convert them into those drop-in chemicals and fuels. And I'll highlight some of the examples here, but really quickly, not all of these examples. Take a deep breath. And I've only got 51 seconds left, so it's almost over. But I wanted to highlight that this is a central metabolic map in E. coli that demonstrates the variety of products that are produced naturally. And so the question is not what can we produce, but what should we produce? And so what we really want is BioShack, right? Another message I want to convey to you is that we really want to establish a BioShack where we can assemble our motherboards and our refineries in a very systematic fashion. And one of the first examples that we did at the Joint Bioenergy Institute is make E. coli that could secrete biodiesel in the form of these fatty acid ethyl esters. And we did that by a relatively simple process of knocking out one enzyme and overexpressing the production of these fatty acid ethyl esters. Okay, I was wondering what was everybody applauding for. We're also doing methyl ketones in E. coli as another diesel surrogate. And I also just wanted to end with that there are other projects related to renewable energy in the Bay Area that are great. And they represent the whole full spectrum of solutions, and thank you very much.
That was excellent, excellent start. So do we have any folks who need to be seated now on this break? Is anyone waiting out there? No? Okay. All right, so next up is Aindrila Mukhopadhyay, who is going to speak about a different kind of microbe or a different use of microbes. Please come up.
Let's see. So my name is Aindrila. I work at the Joint Bioenergy Institute also. I'm a staff scientist at the Berkeley lab, and I'm going to tell you some more about microbes. Now, that last talk was a great example of how microbes are part of these core technologies that are being developed to meet some of the broadest demands of our times and the demands that we are making on our planet. But in my talk, I'm going to talk about a slightly different kind of microbes, and I'm going to open with a very familiar image. This is an image from the 2010 Gulf oil spill. It's possibly one of the biggest man-made disasters. And for weeks afterwards, we thought we had destroyed that entire ecosystem. And we actually had no clue about how to mitigate this problem. Now, a couple of years later, much of that oil is gone. And a very powerful metabolism of this organism that exists in that environment had a lot to do with mitigation of this disaster. Alkanivorax borcumensis is a very famous microbe now, and there are a lot of people studying it, trying to understand why it does what it does and when it will do what it does. But this is hardly the only organism that is our secret friend in the environment. So I'm showing you some images from Arizona from 2012, last year, where there were these horrible dust storms, completely brought life to a standstill for a lot of people. And this primarily happened because we destroyed this beautiful, elegant cyanobacteria that form these mantles over the desert holding it down. These are biological soil crusts. They were alive, and we did a lot of damage in the desert causing these dust storms. You don't even have to go that far. Every protein in all your bodies and every organism that you know of contains at least one sulfur atom. And the sulfur is made available to us by the biological activity of these little microbes, such as Desulfovibrio vulgaris. Vulgaris is the Latin for common. They're very, very common. They're all over the place. And their role in our lives is uncontroversial. The same chemistry that Desulfovibrio uses to convert sulfates to sulfide is the chemistry one would use to convert heavy metals from its oxidized states, where it's soluble in water, to its reduced form, where it's no longer soluble in water. And why is this important? Because it allows you to biocontain this very, very toxic waste that we generate in our energy and nuclear and industrial practices. And it has profound implications on how we can mitigate damage in our environment and how we can live in harmony with our environments. So we've gotten very good. We've gotten very good at identifying who our friends are. But can we always understand what they will do, and can we predict how they will behave in these environments? The environment is a very, very complex place with a lot of different parameters that are dictating the behavior of these organisms. The organisms are not much simpler either. I'm showing you a little schematic of a very small part of Desulfovibrio, and it's already pretty complicated. So you're at a place where you're trying to predict all these complicated parameters coming together and cross-secting with your microbe, and you're trying to figure out how they operate. Can we really do that? Do we know when they will behave the way we want them to behave? Well, survival in the environment, perceiving signals, is not something that only microbes do. We do it too, every day. That's why our species is here today. And at the end of the day, these parameters, these stimuli from our environment, being able to see, hear, touch our environments and respond in the correct manner, boils down to molecular mechanisms. And that is not much different for microbes either. They too have signaling systems that tells them what is happening in the environment so that they can figure out how their response would give them the fitness that they need to survive that environment. Just that in the case of microbes, it's a little bit immediate. They don't have the hierarchy of sophisticated vision or hearing mechanisms. It's very immediate. They have to see the signal, and they have to come up with the response immediately. Most microbes have signaling systems that basically are present on the cell surface that they use to sense their external stimuli and then orchestrate their response. And their response is going to be sort of an aggregate of all the stimuli that they are perceiving. So that's good. We know a lot about these signaling systems and a lot of pathogenic bacteria because we've been studying them for a long time. But what about these environmental bacteria? Do we know? Do we know anything about them? Well, to give you an example of D. sulfovibrio vulgaris, my favorite organism, it was discovered in 1940, maybe 30 years before I was born. Its genome sequence has been known for the last 10 years. And astoundingly, even though it is the focus of a lot of different studies that focus on understanding how it will respond in the environment, applications designed around its activity, its signaling systems, until very recently, were completely unknown. So this is the knowledge gap that we are trying to bridge here. This is the gap in knowing what it can do and what it will do. To be able to predict this is the thought of the day. Any one of these signaling systems, any one of them could be something someone like me could study my entire life and still not understand fully. So we simply don't have to have the time to study them one at a time. So what we did is we devised a strategy to figure out as much as we could about these signaling systems in one fell swoop. And in 2 years, we managed to predict all the genes that are controlled by all the signaling systems in D. sulfovibrio. What that does is gives us connection between the environment and this organism. We can now know that the carbon utilization of this organism relies not only on the ability of this organism to sense whether carbon is present or not, but also on nitrate stress, phosphate stress, oxidative stress, and 2 other unknown stresses we quite haven't figured out yet. What this gives us is an ability to know what the environment means to this organism. What is the connection? How does it see it and its environment? And when something dramatic changes in that environment, how will it respond? Now, in our lives and in our beliefs, we always talk about living in harmony with our environment. As our demands on our planets increase exponentially, it's very important that we understand how our secret allies are in the environment are behaving so as to be able to predict their response. And of course, since I didn't do this work alone, this is my team, and thank you very much.
Thank you, Aindrila. This is going to be hard. This is really hard. So our next presenter is Bill Jagus, who's going to be talking about a different kind of prediction for something we all need to know more about, and that is cognitive decline.
You guys are a tough audience. I'm worried, but I'm going to do it. I've already used up 5 seconds, 6 seconds. Oh, man. Is it crazy to think that we can treat a disease before it happens? No, it's not crazy, right? We do that all the time. So heart disease is the best one, right? Many of you get exercise, not because you like it, but because you think it's good for you. You eat well, and some of you are taking drugs to lower your blood pressure or your cholesterol, all because we think these things are going to prevent heart disease and stroke. And this is an idea that in thinking about the brain, we don't really think it's possible. But I'm going to show you that over the last 10 years, we've had a really different way of thinking about Alzheimer's disease. And through advances in molecular biology and imaging, we sort of have the ability to start thinking about preventing it before it happens. So before I get into this too far, I just want to make sure we're on the same page. Dementia is a term that is sort of a global term for progressive loss of cognitive ability, and Alzheimer's disease is one cause of dementia. It's the disease that's the most common cause of dementia, and I think many of you know that it begins usually with memory loss, and of course all of us are worried about that because we're all forgetting things all the time. So when we look at the brain of a person with Alzheimer's disease, this is a photomicrograph that shows you the plaque and the tangle. And there are two kinds of abnormalities here. There's the amyloid plaque, or made of this protein called A-beta, which is this big, dense, gooey protein that kind of aggregates in the brain. And then there's the tau, made out of the neurofibrillary tangle here, which is made out of a protein called tau. And we have this plaque protein and the tau protein, and they exist in the brain of people with Alzheimer's disease. And by studying transgenic animals and patients over the course of the last 20 years, we've elucidated sort of a pathway by which these proteins get deposited in the brain. And the amyloid protein is actually cleaved. So this is the A-beta amyloid protein that's cleaved from this much larger protein by these two enzymes called beta and gamma secretase. And when they cleave this protein, the amyloid floats off into the brain and forms these plaques. And we think that these plaques may then subsequently actually precipitate the development of this neurofibrillary tangle. So there's a lot of evidence that this A-beta plaque, if it's not the cause of Alzheimer's disease, and we don't really know what the cause is, but if it's not the cause, it's at least a very, very early event in the formation of this particular disease, in the pathogenesis of the disease. So we can now image this amyloid protein in the brain, and this is really the most remarkable development in my career. This is the way this technique came about was by starting with this dye called thioflavin T, and this thioflavin T could be used in tissue sections, just like the one I just showed you, to stain the amyloid protein so you could see it under the microscope. So this is a microscope slide showing you the amyloid plaques stained with this thioflavin T dye. Well, theoretically, all you would have to do would be to figure out a way to visualize this particular dye when it's injected in the brain, into the blood vessels of a person. It would circulate around, just a very simple injection. But it really was a very complicated problem, and a couple of guys at Pittsburgh actually worked this process out, and they figured out that they could label this with a carbon-11, which was a positron-emitting radioisotope. We have a cyclotron in Berkeley that we can use to make things like this. Carbon-11 has a 20-minute half-life. So what we do in my lab is we make this carbon-11 labeled compound, inject it into a person, and then we can image the brain and actually see the amount of amyloid. So we've taken this molecule that's a dye and engineered it so that it gets into the brain with a simple injection, a very low dose of radioactivity to individuals, and we can actually see this amyloid protein in the brain. And this is an example of an MRI scan and an amyloid image scan shown superimposed on each other. And this red is the area of the brain where the amyloid is deposited. And what's remarkable about this particular image is it comes from a normal older person. This is a person who doesn't have symptoms. It's a person who doesn't complain of memory loss, who tests completely normally on memory tests. In fact, this is a volunteer from the Berkeley community. I'm going to tell you, maybe one of you. I hope not. But we don't actually know what this means. We don't know that this means this person will develop Alzheimer's disease. All we know is that this person has this amyloid in the brain. But we think it's a risk for getting Alzheimer's disease. And as I said, it turns out that about 30% of healthy older people in their 70s or 80s will have a brain scan that looks like this. So by studying people who are healthy and following them over time and doing other kinds of scans like MRI scans and all kinds of other imaging studies, we've kind of formed a view of the idea that Alzheimer's disease, the amyloid is there for a long time before symptoms occur. And we think it begins with this amyloid in the brain, and then it develops on to showing degeneration of the brain, where we start to see these changes like we talked about with the tau pathology and the tangles. We start to see evidence of functional change, and we can detect that in other kinds of brain scans. And it's only after a long time, we think probably 15 or 20 years, that we end up with individuals who develop decline in their thinking abilities and dementia. So this is a long process, and in between, we have these other abnormalities that we think the amyloid provokes. Okay, so far, that's all bad news, isn't it? Well, let's go back to this model. And what you remember is that this amyloid protein is cleaved by these beta and gamma secretases, and now we can actually develop drugs that target various aspects of this. And so a number of different laboratories and companies have developed these approaches. One is where we use antibodies to actually lower the amyloid in the brain. The other is where we use enzyme inhibitors that actually block these secretase enzymes and prevent this A-beta from being cut from the larger molecule in the first place. So in theory, we can use these approaches. Now, they've been tried in people with Alzheimer's disease. And here's an example of a study that was published a few years ago, where they gave Alzheimer's disease patients this drug called bapineuzumab. Say that 10 times fast. They gave this drug, bapineuzumab, which is an antibody that lowers amyloid. So did it work? Did it lower amyloid? Well, here's two individual people. This is a baseline scan, and this is a scan after treatment. And you can see the amyloid has gone down in the image. And here the amyloid has gone from this to this. So the amyloid seems like it's been lowered in contrast to the individual on placebo. But don't take two images for it. Here's the aggregate data that shows the placebo group is sort of going up, and the drug group is going down. So it lowered amyloid, but they didn't get any better. And the reason they didn't get any better, we think, is because by the time people get Alzheimer's disease, their brain has been seriously damaged and seriously ravaged. And so the approach now is to identify people before they get sick and treat them with the drug. So the idea is to assess people for their risk. Maybe their age is good enough, maybe genetics. Do the amyloid PET scan, select individuals based on an amyloid scan who would be tried, who would be tested on amyloid with amyloid-lowering drugs, and then see if these amyloid-lowering drugs have an effect, and we can monitor the effects with this amyloid therapy. And these are the folks who've contributed to my research by participating.
So I sense a weakening resolve at the 7:45 mark. You don't want to interrupt these people. So next up, a change of pace. Greg Bell is the next presenter. He'll be talking about scientific networking and big data.
Thanks, Jeff. Hello, thanks. So my idea is that big data is going to transform scientific discovery. You may have heard about big data, and you may have wondered exactly what that means. So let's start with an example, and this is not in a scientific context, it's more in an ordinary context. Imagine you come across a postcard, an older postcard. It has this image on it, an old photo, with no attribution information at all. You have no idea where this was taken, but you'd like to find out. What strategy could you adopt to find that out? Well, you could carry the postcard with you every day, and you could show it to all your friends, and you could ask if they know where it was taken. That might or might not work. Or you could use the internet. There's a new service, I think it's little known, a Google service called Search by Image, and it lets you take an image that Google's never seen before and upload it to the website. And I did that just a couple days ago with this image, which I happened to have taken on vacation. I uploaded it to Google, and Google thought about it for about a third of a second, and it produced an amazing amount of information. It told me what city it was taken in, Istanbul, what structure it was taken in, and what room within the structure it was taken in. Then it provided me with 6 or 8 images that were visually similar, many of which were taken in the same room, and one of which is almost identical to the image that I uploaded. It was taken by a different tourist on a different day with a different camera, and it was posted in a Brazilian website. So this is pretty amazing. It looks like something of a miracle, but behind the scenes there's a lot of infrastructure and capabilities that make this possible. For one, Google owns a global network of massive data centers. This is their data center outside of Atlanta, stuffed full of high-performance computers and storage and high-performance networking. And then there's a small army of experts, applied mathematicians and computer programmers, computational scientists, experts in computer vision. And all this together forms a kind of environment for discovery. And that's what we mean by big data. The data itself is at the center of that environment. And the fact that it's big may not even be the most challenging thing about it. Sometimes it's challenging because it's complex, or it's heterogeneous, or it's very noisy. But that data sits at the center, and all these other capabilities I talked about are combined into a larger discovery environment that lets us answer questions we couldn't answer before. Now, this is what Google is doing. It's what Facebook and Netflix and Amazon are all doing. And increasingly, it is what scientists are doing. And in fact, many scientific fields have been transformed or are in the process of being transformed by this big data paradigm. And climate modeling is a good example. There are a number of examples in high energy physics as well. And in fact, the amazing announcement last summer about discovery for evidence of the Higgs boson that you may have heard about, and Berkeley Lab played a big role in that, was a triumph of big data analysis. And one way to visualize the impact of big data on the sciences is actually to look at computer networks, where all the data has to cross the networks to get from one location to another. And you might be surprised to learn that Berkeley Lab manages a very large-scale computer network. It's the fastest science network in the world. It interconnects all the national labs at national scale. And if you look at the history of data on that network, which is called the Energy Sciences Network, or ESNet, it's really skyrocketing. And that's the impact of big data science. And just a tiny digression on networking, because I manage the Energy Sciences Network, I want to say that these big data flows we describe as elephants, because they are 1 million times or maybe 10 million times larger than the more ordinary flows that you and I generate when we check our email, or we browse the web, or we use YouTube. We refer to those as mice. And it turns out the mice and the elephants in this realm, as in the natural realm, at least mythically, they don't interact very well in the computer networking realm. And in particular, the mice disturb the elephants. The elephants are very sensitive. So we have to build sort of fast lanes for the elephants in our network that can accommodate the big data flows and make sure they can flow at maximum speed. Okay, so where is all this data coming from? A lot of it is coming from massive scientific instruments. This is a picture of the ATLAS detector at CERN, and it's one of the 2 instruments responsible for collecting evidence that led to the discovery of the Higgs boson. And it is one of the largest and most complex and expensive scientific instruments ever built. And it produces a lot of big data. Here's another instrument. This one's just on the drawing boards. It hasn't even been built yet. It's a distributed radio telescope called the Square Kilometre Array, and it will be built in Australia and in southern Africa. And it has hundreds and hundreds of dishes and sensor arrays, and the data that's coming from those parts together adds up to much more data than can be carried today by the entire global internet, in fact, 100 times more. So some of these big instruments are truly producing just massive amounts of data. But in addition to that, smaller instruments are beginning to produce a lot of data. So small environmental or weather sensors, or desktop DNA sequencers, or medical imaging devices, or little individual detectors that are part of larger facilities, they're all producing a lot of data. And that is truly transforming scientific discovery. So back to this picture. You may wonder, how do scientists interact productively with all this data? Is there a tool like Google that enables them to short-circuit a lot of painful manual analysis and generate sort of leaps of insight quickly? And the answer is that yes, those tools are being developed. Some of them are being developed at Berkeley Lab or with close participation at Berkeley Lab, and I'll tell you about just a couple of them briefly. Both of the ones I'm going to tell you about, you can Google and learn about, and I think you can access them online and get your own accounts and interact with them, actually. So they're to some extent public tools. This one is called the Materials Project. It's a tool for materials scientists, and it lets them, materials scientists can use this tool to find new compounds and better compounds for building new batteries or solar cells or many other devices. And they don't need to do that experimentally. They don't need to do it by building or synthesizing the compounds in the lab. They can do it computationally. And this has a couple of advantages. It can speed up their work a lot, and it also holds the promise of being able to let them do things they would never imagine doing in the lab. Here's another example. This is called the knowledge base, or K-base, for predictive biology. This is quite an interesting tool because it brings together over 900 separate data sets into a very rich environment that scientists can use for interacting with the data. This is only one tool, the sort of pathways tool of a couple of dozen that K-base provides. And this tool lets scientists pose questions about and explore the structure and function of proteins and genes. Okay, I want to close with one final idea, which is that Berkeley Lab is really a world pioneer in harnessing big data for science. And that's, I think, because we have most of the ingredients required to create that environment for discovery. So we've got lots of instruments, big and small, that produce a lot of data. We've got one of the world's foremost computational centers, NERSC, Supercomputing Center, currently at Oakland, moving soon back uphill to Berkeley. And the importance of NERSC is not just that it's got a top 5 supercomputer, but it's got lots of experts and staff expert in making that computer useful in a scientific context. We've got an amazing array of applied mathematicians, computational scientists, programmers. We've got the world's fastest science network, and then remarkable domain scientists, physicists, biologists, chemists, like the people I'm sharing the stage with. And together, this creates a really productive environment for discovery around big data. So in closing, the big idea is big data will transform science, and it's happening at Berkeley Lab today. Thank you.
Way to go. So apart from beating the clock, we just give an extra round of applause. You'll note in Greg's presentation how many times he said Berkeley Lab. And that's a very conscious decision on our part, because Berkeley Lab is a name that we're using a lot these days, and we're doing it for a lot of reasons. One is to distinguish ourselves from our other institution that's a little farther east from us, because we found that a lot of people don't know that Berkeley Lab even exists. So that's the name we're using. You're going to hear a lot of it for many months to come. So next presenter will be David Schlegel, who's going to talk to us about the mysteries of the universe. And I don't need this mic because I have another kind.
Okay, great. So all the elements in the universe. I was just going to say all the elements in the universe were generated in about the first 3 minutes, so I should be able to do this in 8 minutes, right? Okay, so I'm going to talk about mapping the universe. So this is the big idea. It's not a new idea. It's something that we've been doing as long as humanity can. But making these maps of the universe, we've actually learned a lot about, well, the universe, obviously, but the components of the universe and the physical laws of the universe. And as an example here, I've put up here a picture of the solar system. Just to illustrate as an example, in 1846, the discovery of the planet Neptune was predicted by making maps of the solar system where it just didn't fit the laws of nature that we knew. So we knew either those laws of nature were wrong, or there was a new body out there, and that was Neptune. That's how that was discovered. More recently, fast forward 100 years, was the discovery of dark matter, also from maps of the universe that didn't fit. So this is material in the universe. It's not made out of the protons and neutrons and electrons that form up most of what you're familiar with, but 5 times more common is this dark matter, discovered again by maps of the universe that just didn't fit. Now, in 1998, there was the discovery of dark energy, which is more common than matter or dark matter. Again, maps of the universe that didn't fit. Okay, so what do I mean when I say a map of the universe? Well, here, this is if you just take a picture of the sky. Okay, so this is a picture of the sky from the Hubble Space Telescope. What you see here are stars and galaxies. But this is a 2D projection of the sky. So this is what's easy to map out. But you look at this picture, and you can't actually tell some of these objects, they're only a few light years away, they're stars. Some of these objects are about 11 billion light years away, so near the observable edge of the universe. And it's really hard to tell from this picture which is which. So not a very good map, I would say. To make a good map, meaning a three-dimensional map, we need to take what we call spectra, where we take these pictures of the sky, and then we take the light from each one of these objects and split it up into the constituent wavelengths of the light. And then that gives us enough information that we can then determine what each object is composed of and how far away it is in the universe. And from this, then we construct a 3D map. Okay. But there are a lot of photons coming from the universe. So these maps, if you look up in the sky, in the previous image, there are about 100 billion galaxies that you could see over the sky. Each one of those galaxies has hundreds of billions of stars, and then they're radiating photons to us on Earth. Now this picture here is just to demonstrate what you could do if you could build the perfect detector for all these photons from distant galaxies. So this is if you just go up to kind of a quiet mountaintop somewhere up in Tilden Park, laid down on the ground, and I'd suggest doing this on a moonless night where you're not being contaminated by all the reflected light from the sun. If you did that for a whole night, then there are enough photons, enough light particles hitting your body from all those distant galaxies that you would get enough information to actually construct a 3D map of all of those objects. Well, actually, so I should say you'd have to do this at least 2 nights. Okay, so you do that tonight, and then you should come back in 6 months when the Earth moved to a different part in the solar system, and then make the rest of the sky. And you should also go to the South Pole maybe for a night. Okay, so that would be the perfect detector if you could turn your body into the perfect detector. We don't have that yet. I wish we did. But what we have then are telescopes like, here's the Hubble Space Telescope. It turns out this is not a very good instrument for making these maps of the whole universe. And the reason is this telescope can only see a tiny bit of the sky at once. So it's the field of view that it can see on the sky. It's about the same as if you take an 8-foot-long soda straw and look through it. And then you can move the position of that soda straw every hour and a half. And you can work it out. Then it takes you about, 200,000 years to try to map the sky. So that's not the right answer. So what we've done is we've built a special purpose telescope. This is in Apache Point, New Mexico, where we can see a larger patch of the sky at once. So this can see a patch of sky about 40 times larger than the full moon. And instead of looking at one object at a time, this telescope can look at 1,000 objects at a time. And we do this by taking fiber optic cables and then plugging them into the positions of 1,000 different galaxies at once. And then we observe those for an hour, and then move the telescope and map a different part of the sky. So this little movie here was someone plugging in all these fibers. Unfortunately, they don't actually move quite that fast. That was in fast forward. Okay, so this has been a great way to map the universe, where over the last 5 years now, we've mapped the 3-dimensional positions of 1.5 million galaxies. So this is the premier instrument on the planet right now for making these maps. That's how quickly we can go. So the future is taking that person out of the loop and replacing that with robots that can actually reconfigure the focal plane of telescopes much more quickly. So what we have here is something we're designing right now at Berkeley Lab, where on the back of a large telescope, we'll have 5,000 little robots, each one the size of my thumb, reconfiguring things on the focal plane of this telescope in 60 seconds. And then every 15 minutes, we can point to a different part of the universe. And then this map I was showing you that we've painfully constructed over the last 5 years, we'll be able to do that in 10 nights. Okay, then once we finish this, well, I should say, so far we've only mapped about 0.5% of the observable universe. And I think I have that here somewhere. Yes, here we go. So this is the map of the universe that we've constructed so far. And for those of you, who's already bought tickets for Star Trek on Thursday night? Okay, they'll show something like this somewhere in that movie, but it differs from this in the notable way that this is the real universe that you actually live in. And I'll stop there.
Thank you, David. So our next presenter will be Ron Zuckermann, who's going to be talking to us about mimicking nature. Ron?
Thank you. So let's move now from the expanses of the universe down to the fundamental building blocks of life. So it turns out that the building blocks of life, primarily nucleic acids, proteins, and sugars, all share one thing in common, and that is they're polymers. So polymers, we think of something like paint or glue or plastics. But in biology, polymers do much more interesting things. Nucleic acids store the information of life. Proteins are the machinery of life. And sugars are sort of the energy source of life. So as a material scientist, I look at this and say, gee, what's up with this polymer thing? And furthermore, what is a polymer? And a polymer is a particular structure. It's a linear chain of building blocks strung together like beads on a string. And so why does nature use that type of architecture? And the answer is, I think, is pretty simple, and that is it's about a simple set of building blocks. It's very efficient to build stuff from a common set of building blocks. And so here I'm showing the 20 amino acids that represent all the building blocks you need to make any protein in our body. And the way that nature does this is by stringing them together in a particular sequence. So it's the actual order of monomers in the chain that determines a protein's shape and its function. And essentially, you can think of the sequence as folding instructions that tell the chain how to adopt a particular structure. And so here I'm just showing some of my favorite protein structures, like silk is one. This is a viral capsid. Proteins also do interesting things like bind very specifically, like myoglobin binds oxygen. Here's a DNA binding protein. And proteins also catalyze chemical transformations. So as a material scientist, we want to learn from this blueprint set from nature and understand how to build synthetic materials that are much more stable and rugged and we could use in industrial processes and as sensors and that kind of thing. So what we need to understand then is the relationship of this sequence of monomers to structures like these. And so I liken this to origami. Origami, you take a common starting material, a piece of paper, and by providing very specific instructions, you can achieve all kinds of different shapes, okay? So the key is, if you've tried this, it's actually pretty hard. The key is understanding these folding instructions. And so that's what I'm doing in my research. So here is the chemical structure of a protein, just a short chunk of it we call a peptide. And in my research, here I'm showing the amino acid building blocks. These are all linked together one after the other. And what I invented in my lab are molecules that are very similar but totally synthetic. These are called peptoids. And the cool thing about peptoids, here's the unnatural amino acid unit. These are not degraded by enzymes, so they're much more rugged and stable. So the question is, how do we link these together? How can we make materials out of them? Another neat thing is the chemistry we developed is amenable to using all kinds of different shapes and sizes of these so-called R groups, or the side chains. And we developed synthesis methods that where we use robots to link these units together. So here, showing addition of one of these beads along the chain, and we can make all kinds of different sequences in parallel and then evaluate which ones work the best. So here we're adding one monomer, here's another monomer, and we can do parallel reactions. It takes about an hour to add each building block, and so takes time, and but you can do it the robot can do it while you're home sleeping. So what do we make? Well, so we need to learn from biology. So here's an example of a beautiful protein called green fluorescent protein. It's from jellyfish. It actually glows green. And we can learn from this by looking at it through a special set of glasses, which I will simulate for you. If we consider the building blocks that make up this protein, if we look at this through special glasses that only show you amino acids that like water and amino acids that don't like water. So this idea of oil and water separating from one another is one of the fundamental things. So when we look at it that way, you can see some interesting patterns. So red is hydrophilic, that means it likes to be in contact with water, and yellow is hydrophobic, that means it hates water. And what you'll see is this striping pattern. And what that does, that sort of sequence arrangement, you'll notice puts all the yellow groups in the inside and all the red groups on the outside. And it makes sense. These proteins exist in water. What other way can you hide from water except by clustering into the inside? So we applied this rule to non-natural peptoids. So here I'm just showing the chemical structure where the yellow is the hydrophobic group and the red is the hydrophilic group. So I would call this a twofold repeating pattern. So we made synthetic peptoids with the same exact pattern and asked, well, what's it going to do? What's it going to fold into? And to our surprise, we were not expecting this kind of thing, but we ended up making what we now call nanosheets. It's a very highly ordered material. These very sharp edges, very surprising that these polymers are very flexible, like cooked spaghetti, and yet somehow they recognize each other and form very ordered materials. This is a little bit harder to see, but you can see these by optical microscopy. They're up to about 1/10 of a millimeter in size. It's pretty large. And so here I'm highlighting one nanosheet. And we're wondering, okay, what is the structure of this material? And what can we learn from what we synthesized? And we find that the peptoid chains actually stack up in this brick-like pattern. And they stretch out, and when they meet their neighbors and make these, like floorboards, this highly ordered structure. And so this section of sheet is actually about 800 million peptoids lined up next to each other. And how do we know that? We can see that. So up at Berkeley Lab, we have some of the most powerful electron microscopes in the world. This is at the National Center of Electron Microscopy. And here you can see individual peptoid chains running parallel to each other. This is the very edge of a sheet. And okay, so let me just wrap up by saying it makes sense to hide all these hydrophobic groups in the center. And the last thing is this is a movie now that we're getting our supercomputers and our theory colleagues together to look at actually how these form and use this to engineer future advanced materials. So thank you.
Thanks. I'm going to give this to you now, but wait. So, our last presenter is the legendary cancer researcher Mina Bissell, and she's asked for some additional time. All right, but wait. She says tomorrow is her birthday, so what do you think? Should we give her a little extra time? Okay, we'll do so. Mina, welcome.
Thank you very much. Thank you. So a number of you have heard me. I'm the 4th time in this stage, and of course, 30 years coming to Berkeley Rep. Now, I did not ask for more time. They gave me more time. But I thought I'd give them an excuse to allow me to do this, because I gave a talk. It's known as a TED Talk, and some of you probably have seen it. It's about 16 minutes. I'm going to do it really fast and see how many minutes I can cut. Okay, now, so when your mom and dad meet, and some of you have seen this, you have an egg and a sperm. And you know why we have so many sperms per one egg? Because the guys never know how to ask for direction. So the sperm, one of them finally hits the egg. It's now a fertilized egg. It becomes like this, it becomes like that, and before you know it, it becomes like that. Now, imagine how many cells this woman has in her body. She came from a single cell with the same genetic information. And she has, does anybody want to guess? 10 to 70 trillion cells in her body. And these are more than the national debt of US and Europe put together. Imagine, they all have the same genetic information. Now, have you ever thought about what 70,000 cells look like? The number 70,000? It goes and goes and goes, and this is not doing any of the things I expected it to do. So you have to watch my TED Talk. So we decided that it is really important to find out. It actually, after my old age, you know, I have been doing science for 40 years. This tells me I can never take anything for granted. I should have actually gone and looked at their slides. My assistant sent the slides, and they're all scrambled. So what can I say? All right, here is a human breast. We ask the question of how does this 70 trillion cell, all with the same genes, make your nose, make your finger, make your breast, and for the gentleman to listen, make the prostate. So, how do they do it? And we decided that we really know nothing about this, and what we are going to do is we are going to have to make a model. We chose the mammary gland. That's actually a picture of the human mammary gland, and this is this beautiful three-dimensional structure of one of these things in the human breast, and around it are fat, but we have about 7 or 8 of these. And we decided that even that was too complicated, and what we are going to really concentrate on is to concentrate on what we call an acinus, which is what the mammary gland makes when an animal gets pregnant. You have one layer of beautiful epithelial cells, one layer of what we call myoepithelial cells, and then around it, what we have is something that I knew nothing about, and I had to learn about it. So we have, over the years, my goodness, over the years, asked the question, what actually makes a breast, and why, when you take the cells out of the breast and you put them in the dish, why do they forget where they came from? So here is a picture of a single mammary gland or breast from human breast. You see how beautiful it is? It has very lovely structure. It has a bottom, it has a top, it has a secretory activity. You take these cells and you put them in the dish. In about 2 days, every one of them forget. They were making gobs and gobs of milk. They had fat droplets, they were making the milk coming out, they were doing all sorts of wonderful things. They are polar, they have a bottom, they have a top, and these forget. So it says that the cells, once a nose or once a breast or once a prostate, don't always remain a nose or a prostate. So the question is, what actually gives them the information? What makes them do this, right? So you look, for example, at the nuclei of these. You see this is in the dish, this is in vivo. You put these things in the dish, they forget to make milk. So we ask the question, why is that? So if we do a section of the mammary gland of the mouse, we see that around these acids is this red stuff which is called extracellular matrix. And these extracellular matrix components are like collagen, huge molecule. People believe that these things actually give you structure, and they don't have information and signaling. And I decided that because they lose it, maybe what the cells didn't have in a tissue culture dish was actually this material. So we actually put and found a material that we were able to put these cells in that material. They could bring it around themselves. They could make a three-dimensional structure. In animals, they look like this. In 3D culture, they look like this. And once they find their three-dimensional architecture in this gelatinous extracellular matrix gel, they now are remembering, hey, I'm a mammary gland, I'm a breast, I know what to do. So we believe that the basis of organ specificity in all of you, when you look at yourself as a miracle in the mirror, is the architecture of that organ. There is wisdom in your nose, there is wisdom in your liver, there is wisdom in your breast, and it all comes from the architecture. Now we can make this gorgeous looking thing full of milk inside this three-dimensional structure, and you can imagine I had a picture of me when I had one of these big birthdays with a big mustache of milk, saying, we got milk. Okay, so we can make these lovely structures of human or mouse breast, and we can do all sorts of other things. This was supposed in the beginning of my talk, and it has come now, so I'm not going to pay attention to it, only to tell you that that's one of the most clear examples of why when people get cancer and everybody tells you that they have had a single cancer gene that is going awry and is giving them all these cancer is not correct. You take a cancer gene, this famous virus, you can put it in the chicken and it gives you that tumor, that ugly tumor. You put it in the embryo, which we did. Here it is in this beautiful feather with these blue things, and it turns into a feather. It just finds a different home. So it says that the context in which a cell finds itself is absolutely dominant over your genome. So your genome is not necessarily your destiny. It's a lot of your destiny when you're born. But once you're born, the microenvironment of those genes are dominant over the phenotype. This is really a pity, though. So we made a hypothesis. We said if this architecture is, in fact, the most important thing that happens, we should be able to take a cancer cell, give it the right context, and they should think they're normal. Alternatively, we could take a mouse, destroy the structure without giving them a cancer gene, and they should become tumorigenic. And we have done both of these, and I don't have very much time to tell you, except to show you. Here are the non-malignant cells in an assay or in an experiment that we have developed where they make this beautiful structure. These are the nuclei. This is the cytoskeleton. They make an acinus. Here is malignant cells. And when we took these malignant cells and we measured what was in the surface, we found out that these malignant cells, in fact, did many of the things these do, but they had completely lost the balance of what is at their cell surface. So we took an antibody to one of the molecules that actually signals to the extracellular matrix and brought the level of that molecule to the level of normal. And look what happened. They all became normal, completely turning the cells into a cell that is a structure that is normal. If we put this into the mouse, while these make 100% tumor, the whole structure makes 0 tumor. If we dissociate them and put them with this antibody, then they get 70% less tumor, and it takes for months before they do it. It was very exciting, as you can imagine. And this is the assay we have shown, and we have done this. And you can see here on the left, every one of those are like hundreds and thousands of human tumors. And on the right, these are tumors that got reverted to a normal phenotype. It's absolutely exciting. All right. So the conclusions are that growth and malignant behavior are regulated at the level of the architecture. And I will tell you that you get Alzheimer because the architecture of the brain starts going. And you get problem with mammary gland or the breast because we age. Aging is the biggest risk factor. So form and function are related dynamically and reciprocally. This was a beautiful thing that was going around like this. I have no idea, Jeff, what happened, but honest to God, oh, there it is, look at it. So form and function are related dynamically and reciprocally. You mess up one, you mess the other, is like yin and yang, right? All right. So where do we go now? So the thing that for you to remember is that as you are sitting here, in every one of your 70 trillion cells, this is going on. Every cell has to know where they are, and their context has to be correct. They go from in to out and out to in, and I call this the model of dynamic reciprocity. Okay, now, look at this. They made this discovery very recently, last year, where you put one of these things in a gel. This is human breast adult cell. In order to make that structure, we discovered a whole new movement. It does this incredible stuff that you just saw, and we have a very beautiful paper that shows that when the normal cells know what to do, the malignant cells don't. But when we revert them, they attach to each other, and they start doing this movement again, so they make the correct scaffold on which we are now able to build that structure again. And this is going to be very important in bioengineering human tissues. So I want to finish by a poem that I love from Yeats. I always debated, do I want to be an English major or do I want to be a chemistry major? But at the end, science won, but I love the literature. He says, "Oh, chestnut tree, great-rooted blossomer," never mind. Here is the important thing. How can we tell the dancer from the dance? It's among the school children. How do you tell the dancer from the dance? Here it is, this beautiful Merce Cunningham, with whom I took some lessons. And as you see, when he's dancing, he's a dancer. When he stops, he's neither a dancer nor we have a dance. So is with form and function. So is with the structure of your nose and the structure of your mouth and why they don't turn into each other, thank goodness. But at one point, somebody took a cell from a sheep called Dolly, right? It came from a mammary gland. So that cell in the mammary gland had all the information to make all that sheep. So you can imagine that we could one morning get up and have two heads, and thank goodness we don't. But if you... So here is my group, and that lovely gal in here was my physicist who discovered that particular movement, and I like to say she was in the right context. She came from Trinidad. And her father was an engineer, her mom was a mathematician, and when she was born, they said, "You're going to be a physicist, you're going to be a physicist, She's a brilliant physicist. And it's not because physicists are that much smarter. We have bad physicists, I know. But she is brilliant, and I have had 3 other postdocs who were physicists. So context determines everything, including you and me, including how we respond to each other. Well, this is my favorite cartoon. So I used to be the cat, and this was the authorities, right? And the authorities said, don't you dare think outside the box. I couldn't get money. People thought I was crazy. And now I always lecture to the young people and say, always think outside the box. Always think outside the box. You have only one life. Live it well and do something good. And I'm proud to be a member of Lawrence Berkeley Lab. It's a great place to be in. Thank you.
Okay, let's start our questions. Looks like we have one right here.
Okay. First, I've got a question for Blake Simmons about biomass. It will save us energy if we're not, money if we're not importing a bunch of oil, but if plants and sugar have carbon in them, then how is that going to be an advantage in terms of climate change if we're just burning fuels that are made from things that have carbon in them? And then another really quick question for Bill Jagast. How can I sign up for a clinical trial to get a PET scan and receive treatment to prevent Alzheimer's? I'm ready.
All right, let's start with the first one, Mr. Go ahead.
So, first of all, I agree with you. I was going to ask the same question about the PET scan. I think that's a great opportunity. But that's a really good question about the carbon balance between what is the current ground state and where we want to go from the renewable economy. And there are many different ways to answer it. But the bottom line is that we really want to close or establish a zero carbon balance from the renewable sources, gas compared to petroleum and fossil sources. So right now, we pull stuff out of the ground. It's very economical, it's very cheap, and we burn it, and the carbon goes up into the atmosphere. And then the process by which you could sequester that carbon and put it back into plants or humans or other uses of it, the time interval is really long, right? So what we want to do, or what is proposed, is that we can take these renewable sources of carbon, meaning that they can be generated on a length scale or a time scale that is of interest to humans. Because none of us have billions of years to wait around for planet Earth to do the carbon cycle again for us. So what we want to do is when we take the carbon and we emit it and we burn it, it gets transformed back into plant biomass or algal biomass or something that can be generated over a course of months to years. And then that becomes our source for those chemicals that we burn. And so it's basically closing that carbon loop where you have a source term, because we're still going to burn it, but now we have a sink term that is much more rapid and much more efficient than relying on terrestrial ecosystem development. Another part of it is by conservation. And that often gets lost in guilt-free America, that we can just keep burning what we want to burn and we'll get better, more efficient engineering from it. So lots of people may or may not know that we now have these new fuel efficiency standards that could drive it to 50 miles per gallon on a single gallon of gas. And that has a huge impact on the amount of petroleum and fossil fuels that we would consume for any sector, especially the light-duty vehicle part of the transportation sector. And so when I put that plot up showing that we could produce 1 billion tons of biomass theoretically, and at today's levels of consumption, that would be 1/3 of the fossil fuels that we consume for the transportation sector. If we achieve those standards, it now becomes a 50 to 60% displacement. Because by that single stroke, by driving better vehicles that are more efficient, improving the engines of tomorrow, and designing the fuels for them as well, we can really make a dent in that carbon cycle and make it much better and much easier to attain the greenhouse gas reduction targets that we've established. If we don't, if we just keep burning, what we create and keep consuming and growing, and this is a big concern in the developing world and the growing economies, we're never going to be able to make it renewable enough to make an impact. And so it has to be by better engines, better fuels, and better land management to make it all work.
Okay, get the second question.
Right. I was going to make a joke and ask you to repeat the question, but but I remember it because I've gotten it before. So a lot of what I told you, the work about that whole idea of how to think about Alzheimer's disease, some of that work was done in Berkeley. But we're not actually going to be doing this clinical trial in Berkeley, and the clinical trial itself hasn't started. It's going to start up around the United States. at medical centers. And so there'll be a very large publicity, a large PR campaign at the rollout of this trial. I will tell you that there will be certain criteria. So as I said, about 30% of older people have these kind of abnormal scans. So we estimate that we want to study 1,000 people, so we'll have to scan 3,000 people, and we'll probably have to screen 5,000 people. So we're going to be looking at people who are most likely to have amyloid in their brain, and that means people who are fairly old, and we may be looking at genetic factors as well. So there'll be a screening process, and then a scanning process, and then people will be put on either a drug or placebo. So it's going to be a clinical trial, so there's a 50% chance that a person would be given a placebo. As I said, we won't be doing the study in Berkeley, probably be at medical centers in the Bay Area, and it'll be rolling out later this year.
Thank you.
Thank you for the questions. Next up right here.
Yes, for Dr. Bissell. I was wondering if you could say more about the clinical implications and the clinical developments that have been based on what you found about the environment for the cells.
Yeah. Let's see if can you hear me? Is that on? Yeah. Okay. So one of the big things that I didn't get to elaborate on is the discovery that we have made is that the tumor itself could have a bad genetics, but the microenvironment around the tumor also determines what the tumor does. And one of the reasons we have not succeeded in doing in cancer a lot better than we should, considering that is one of the few areas where we have spent a lot of time and a lot of money, etc., is because we have not paid attention to the microenvironment. Because we can show that when we mess up the microenvironment, actually tumor starts growing. And if we can do something good about the microenvironment, we can stop a lot of things. And part of it, of course, is prevention, but the other part of it is to treat both the tumor and the microenvironment. And that's why exercise is so good for you, because people who have tumors and have had cancer, if they exercise, they get much, much less relapse. The metastasis doesn't occur as often. And also, if we are able to correct and do something with the microenvironment, like stop the inflammatory response of the person who has had it, plus removing the tumor, it does a lot of good. And I now stop so other questions could be asked, but at the end of my talk, I can also tell you some other stuff we have done about metastases and dormancy, because after all, metastasis is what kills people. And we have a fantastic way of dealing with that now.
Yes, we have a question right here.
Are the engineered proteins biodegradable?
Yeah, that's a great question. The ones that we make are not biodegradable, so they you know, they do not degrade. However, there are a number of cases where that's actually really important. So, for example, if you want a diagnostic test for a disease, and you want to put a drop of blood on it, a lot of the things that we normally would think of to use do get degraded, and so they don't last, and they don't make a reliable, robust test. Or, for example, if you want to test for toxins, say to give our soldiers that work in really harsh environments like the desert, and you want diagnostic reagents that are really rugged and don't degrade. At least when we say biodegradable, we're thinking of short time scales. Of course, all the stuff we're working on is eventually will degrade because it's just made of.
Isn't it also important if you are flying on an airplane, you don't want the airplane to biodegrade, right? So think about all these things that we don't want to biodegrade per se. But when we put them someplace and let them rust, then we do something else with them. But these guys come up with the materials for things like airplanes. And it's important that it shouldn't be biodegradable. So it's not one single answer to every question. In some cases, it's very important. All those horrible plastic bags are useless. We don't care if they don't become biodegradable. So if you pick up the green ones, they become biodegradable, and that's good. But if you sit in an airplane, you don't want that to happen.
We have one over here.
This is a question for Dr. Schlegel. If an explanation could be found for the red shift other than constant, nonstop expansion, would we still need a concept of dark energy and dark matter?
So David, be sure to explain the red shift for those who might not know what that is.
Right, okay. So the way that we make these maps of the universe is, all I showed up here was I said we take a spectrum of these objects and then we use that to determine the distance of an object. That's by measuring its redshift, or the Doppler shift of the light. And then we use that combined with our knowledge that the universe is expanding to then place the objects that are moving more quickly further away. So that's presuming that we can do this mapping of redshift or Doppler shift to distance. So I should say, if you go back, at one point this wasn't well established, but there are many other pieces of indications showing that the general framework for our picture of the universe is very well understood now. So the fact that we did have the Big Bang, that it was shortly after the Big Bang that the elements formed in the cooling universe, and then we can see the distant universe actually looks nothing like the universe does today. So if we look at galaxies that we see that are halfway across the universe, or 3/4 of the way across the universe, or even 80% of the way across the universe, we're seeing snapshots of the universe when it was very different, and we can tell that the universe was indeed very much younger then. So that if we look at objects that are at what we say a redshift of 2, meaning that the light's been stretched out by factors of several, we can tell that those objects, none of them are very old. They're only 1 or 2 billion years old, as compared to, we look locally, and well, the sun is 5 billion years old, and there are other stars near us that are even older than that.
Thank you. Next question.
This is for Dr. Jagast. In addition to the method that you explained of treating Alzheimer's, in which you prevent the cleavage of A beta, would it also be possible to treat or delay Alzheimer's by adding more adenyl cyclase enzymes, or by lowering the breakdown of cAMP?
Well, so those are signaling pathways within cells, and it's not clear exactly how the amyloid itself does its toxic damage. So I guess I can't really answer your question because we don't really know well enough how the amyloid damages the cell. But one of the things that it does, for example, is it affects the neuron in terms of how the synaptic function works and how the signal from at the synapse is sort of transduced into the cell. So definitely things like cyclic AMP play roles in that, but there's really no good solid evidence that would be a pathway to treat for Alzheimer's disease at this point.
Thank you for the question. So before we take our last question, I was going to ask Greg and Aindrila, considering that you guys finished close to your time, if there's any single point you want to add that I didn't let you add because of the time limit. Was there anything that comes to mind?
I got everything in.
You got everything in? All right.
Just barely.
Is there anything, Aindrila, that you would like to add to yours?
I'd just like to add that perhaps all the work that you hear here are actually interrelated.
Very good.
I'd be a little hard-pressed to add in the dark matter or the dark energy. But aside from that, it's actually all about balance and sustainability, whether we are talking about the microenvironment around malignant tissue versus a normal tissue, or our policies regarding how we consume our materials. You showed these beautiful images of futuristic Australia, where they're putting these huge data gathering systems in the desert. I got the sense that there were some some mistake that the deserts were a great place to put it. Because in fact, those deserts are alive. And if there are not any policies in place to figure out what will happen to the ecosystem of that desert, when you put in that kind of structure, you're going to have a whole other kind of problem to mitigate. So there is this sense of big science and broad models that take a more global view of any solution and realize that it is not a silver bullet. Every problem we try to solve comes up with a whole new problem, and unless we understand what we are asking, we'll just create more problems.
Great, excellent point. So you have the honor to ask the last question of the evening.
Okay, thank you. So this question is going to be for Dr. Jagas. From my biochemistry classes in undergrad, I remember that the beta amyloid precursor protein was actually cut in a different order by another. It wasn't beta or gamma secretase, probably the one in the middle. And so I'm trying to think why the antibody therapy against amyloid beta, also the beta secretase inhibitors, haven't worked. And could it be because it's a ratio between the good and the amyloid beta, which just accumulates and forms plaques sort of as like a side effect? Because I know that neurons do have some wound healing properties, so it can't just be that, you know, when the plaques are gone, they should be able to repair and restore at least a little bit. And so what do you think about that?
Well, you know, the brain's ability to restore and repair is really very limited. There's really only a few areas in the brain that do what we call neurogenesis. Mm-hmm. And if you look in the brain of a person with Alzheimer's disease, it's just vastly disturbed, vastly damaged, way more than those parts of the brain that can actually generate new neurons. So the ability, plus the older we get, the harder it is to generate neurons, and especially you don't just generate the neurons, you have to generate the appropriate connections. And those connections are formed over the life, especially when you're young. Those connections are really formed in youth during development. So I think the process of hoping that neurogenesis or stem cells, for example, is going to solve the problem, it's certainly a reasonable approach, but we're very, very far away from that. I think we're further away from neurogenesis or stem cell therapy than we are from the idea that maybe we could prevent this whole process in the beginning. But they're both approaches that we have to consider.
Okay, thank you.
Thank you for the question. So please tell your friends about Berkeley Lab. Word of mouth is very important. Please tell them. I hope you've enjoyed this evening. This concludes our presentation. Thank you, scientists, and thank you, audience, for your applause. The video will be available on our website by tomorrow, we hope, and then there'll be a broadcast quality one that comes later. And those of you who are friends of Berkeley Lab, please look for information about our fall series, which will begin on September 9th. Thank you all for coming.